In this video, we are going to be reviewing the entire process of protein synthesis, starting with transcription. This is kind of step one of the two-step process. If you recall from the presentation, this occurs in the nucleus. Starts with, and this is our cell here, in case you were looking at my abstract drawing over here and wondering what exactly it was. This is our cell, and then this structure inside the cell is the nucleus, and then this little twisty ladder thing in here is DNA. In transcription, the DNA is going to separate. So here we have our DNA. Our DNA kind of zoomed in. Notice the A's, T's, C's, and G's. And then we have the mRNA here coming in and binding with the DNA. So again, DNA is in green, mRNA in red. And here is our DNA. We call this the template strand. This is what has the specific gene in it. And we're going to use that to make a protein. But we have to get that message out, and that is what the mRNA is going to do. So in making our mRNA, T binds with A binds with U. Remember, RNA does not have T. So in our mRNA, you're not going to see any T's. G's binds with C's, C and G, T binds with A binds with U, T binds with A. And then the mRNA leaves the nucleus. And here we are at step 2, translation. This happens in the cytoplasm on a ribosome. So the mRNA is going to find a ribosome. Again, the ribosomes are free-floating or bound within the cell. And once the mRNA finds a ribosome, here we have our mRNA. And then in yellow out here, this is our ribosome. We have our tRNA here that comes in and binds with the mRNA. But it doesn't just come in and bind, it brings with us an amino acid. And if you recall, amino acids are the little pieces that make up a protein. Okay, so the tRNA brings in the amino acids and they get strung together to form a protein, also known as a polypeptide. So in terms of the bigger picture here, we're going to put it all together. Give me just 1 second. And I'm switching over here to this document. This is kind of a nice picture overview of protein synthesis. If you haven't already written this down in some capacity, I feel like you should, but it is totally up to you. Here I've outlined the concept, DNA to RNA to proteins, which is what we call the central dogma in biology. Remember, DNA is double-stranded, has the bases A, T, C, and G, and RNA is single-stranded, A, U, C, and G. And that message of DNA stored in RNA is the process of transcription, and that happens in the nucleus. Here we see in this picture, the mRNA is coming out. This thing over here is the nucleus. That mRNA, it's kind of like a ribbon, threads through this thing here, the ribosome. And now translation can begin. Again, this is in the cytoplasm. Here we have our tRNAs, and then attached to the tRNA is our amino acids. So these are the amino acids. The tRNA is here. And on the mRNA, there are these little groups of 3. And these little groups of 3 are called codons. So the group of 3 mRNA bases tells you what amino acid is going to be attached to the tRNA. And that's kind of what we call reading the genetic code. And I have a video all about that. And we can see in this next scanned document here. A little bit more detail here in terms of the codon. This is our codon here, and that codon reads GCU. GCU is genetic code for the amino acid alanine. And alanine is brought over by our tRNA. It gets attached to other amino acids to create the polypeptide chain, also known as a protein. Just piggybacking off of that, GGU is genetic code for the amino acid glycine, and it's the tRNA here that brings over the glycine. And that is a summary of protein synthesis, specifically some more details about transcription and translation. And thanks.